All right, welcome back. Today, we're diving deep into one of the most compelling areas of Parkinson's research, how specific environmental toxins can actually kickstart the whole cascade of events that leads to neurodegeneration. We're going to peel back the layers here, going from the big picture studies right down to the molecular nuts and bolts of how these chemicals target and destroy the very neurons we need for motor control.
Okay, so here's the roadmap for our explainer. We're going to start with the central mystery of Parkinson's itself. Then we'll look at a really tragic accident that, believe it or not, give us a massive clue. From there, we'll see how that clue connects to common pesticides. Then we'll zoom right into the cellular machinery they attack. And finally, we'll tie it all together by looking at how our own genes play a role in all of this.
So before we can really get into the nitty-gritty of these toxins, we first have to get a handle on the central puzzle that's been driving researchers for decades.
And this right here is the core mystery. See, Parkinson's isn't a disease that affects the whole brain uniformly. It is laser-focused on the profound loss of a very specific group of neurons in a part of the brain called the substantia nigra. These are our dopamine factories, and when they die off, we get those classic motor symptoms. So the billion-dollar question is, why them? What makes them so uniquely vulnerable?
Well, it turns out there's probably no single, simple answer. The consensus now is that Parkinson's is a multifactorial disease. Think of it like a perfect storm. An individual's genetic code might make them a bit more susceptible, and then an environmental trigger, like one of these neurotoxins, comes along and pulls the trigger. These two forces then converge on the same pathways of damage inside the cell. Things like massive oxidative stress, which you can think of as cellular rust, and a breakdown in the cell's energy factories, which we call mitochondrial dysfunction.
All right, now for a really pivotal moment in this whole story. And it's one that came about not from a carefully planned experiment, but completely by accident, out of a public health crisis.
So picture this. It's the early 1980s, and a group of young drug users in California suddenly developed these incredibly severe, irreversible symptoms of Parkinson's. It was baffling. The cause was eventually traced back to a contaminant called MPTP in a bad batch of synthetic heroin. It was a tragedy, for sure, but it provided researchers with a smoking gun, the first undeniable proof that a single chemical could produce the exact pathology of Parkinson's. This gave them an incredibly powerful tool to finally model the disease in the lab. And this is where we see just how specific and, honestly, kind of clever this toxin is. First, because MPTP loves fat, it just slips right through the protective blood-brain barrier. But once it's inside, it's not even toxic yet. It gets converted into its deadly form, MPP+, by an enzyme in neighboring cells. And here's the kicker, the key to its specificity. MPP+ looks so much like dopamine that it tricks the dopamine transporter, the DAT, into vacuuming it up. It basically has a VIP pass right into the dopamine neurons. Once inside, it makes a beeline for the mitochondria, where it slams the brakes on a crucial enzyme called complex 1, crippling energy production and ultimately killing the cell. So the MPTP story was a total game changer, because it immediately raised this really unsettling question. If a rare synthetic chemical can do this, what about more common chemicals that we're exposed to all the time?
And that's exactly where the research went next. The focus just blew wide open. Scientists started to wonder if widely used agricultural chemicals, some of which looked a little bit like MPTP, could be a factor in what we call idiopathic Parkinson's disease, which is really just the term for cases where we don't know the cause. Two big names popped up on the radar almost immediately. Rotenone, which is a pesticide that's sometimes even used in organic farming, and paraquat, a super common commercial herbicide. And look, this wasn't just some theory cooked up in a lab. Multiple large-scale studies of human populations started to draw a clear statistical line connecting long-term exposure to these chemicals, often linked to farming or just living in a rural area, to a significantly higher risk of developing Parkinson's.
Okay, so we've got our suspects, but how do they actually pull off the crime? To figure that out, we have to shrink down, way down, to the subcellular level, to the scene of the crime inside the neuron, the mitochondria. Right, so we're inside the mitochondrion, the absolute powerhouse of the cell, and its main engine is something called the electron transport chain, or ETC. Think of it as a biological assembly line. This series of protein complexes is what generates almost all of the cell's energy. It's ATP. It's absolutely vital for keeping high-energy cells like our neurons alive and firing. And here's the critical piece of the puzzle. Both MPP+, from the MPTP, and the pesticide rotenone, they don't mess around. They go straight for the jugular, launching a direct attack on the very first and biggest part of this energy factory, complex I. By taking out that first step, they effectively shut the entire operation down. And blocking that first step creates this two-pronged catastrophe inside the cell. First, you get an instant energy crisis. ATP levels just plummet, and the neuron is essentially starved for power. But second, you get this massive traffic jam of electrons. They have nowhere to go, so they start to leak out and react with oxygen, creating huge amounts of these incredibly destructive molecules called free radicals, or reactive oxygen species. The whole cell is just flooded with oxidative stress.
So it seems like they all lead to the same disastrous outcome, right? Mitochondrial failure, oxidative stress. But if you look a little closer, you'll see they don't all take the same road to get there. This is so fascinating. What you're looking at here is a classic case of molecular mimicry. I mean, just look at them. On the left is MPP+, that toxic byproduct from the synthetic heroin. And on the right, the super common herbicide paraquat. It's no wonder scientists started looking at paraquat. Their chemical structures are just uncannily similar.
But this is where the story gets a little more complex. Let's break this down. You can see that MPP+ and rotenone are basically sledgehammers. They are potent, powerful inhibitors of complex 1. But paraquat, even though it looks so similar, is actually a really weak inhibitor of complex 1. Its primary weapon is totally different.
Paraquat's main weapon is something else entirely. It's a process called redox cycling. It basically hijacks electrons inside the mitochondrion, maybe at a different spot like complex 3, and uses them to just endlessly churn out massive quantities of free radicals. It becomes a non-stop free radical generating machine. So let me put it this way. For MPP+ and rotenone, that oxidative stress is a byproduct of shutting down the power. But for paraquat, creating that stress is the main attack.
Okay, so this all brings us to the final and maybe the most important piece of the puzzle. Millions of people are exposed to chemicals like these, but obviously not everyone gets Parkinson's. So why not? Well, the answer seems to be in this intricate dance between these environmental toxins and our own unique genetic blueprint.
And that's where this idea of a multiple hit hypothesis comes in. Think of it like this. Exposure to something like a pesticide is the first hit. It's a punch. But whether that punch leads to a knockout, well, that depends on the person's underlying genetic resilience or vulnerability. That's the second hit.
And this isn't just a theory. We have really solid lab evidence to back this up. For instance, if you take mice that have a mutation in a Parkinson's-related gene called DJ1, they are way more susceptible to damage from paraquat than normal mice. Same thing on the other side. Mice that are engineered to have a mutant form of alpha-synuclein, another huge gene in familial Parkinson's, are much more sensitive to MPTP. It's clear proof that our genes can set the stage, basically lowering the amount of toxin it takes to push a neuron over the edge.
And so that leaves us right here, with this huge, critical question that researchers are still tackling. We found some of the keys, right, these environmental toxins that can definitely unlock the door to Parkinson's disease, but the ultimate challenge is figuring out the full combination of locks, the ones encoded by our individual genes, and the sum of everything we're exposed to in a lifetime that determines, who walks away unscathed and whose neurons are ultimately pushed over the edge.